molecules along with the solvent are called selectively, this membrane is called selectively permeable membrane. Basically, cell membrane is selectively permeable. It is not true semi-permeable. Whenever the term semi-permeable is used, it means that it allows only solvent to pass through it. It is also allowing the solvent to pass through it. That's why the term used here is that selectively permeable. Question comes sometimes, what is mean by selective permeability? Selective permeability means the permeability that actually allows the movement of some selective solutes along with the solvent. Living membranes are actually selectively permeable. Third one. It is made of protein, lipid, protein. Protein, lipid, protein means in between two protein layers, a lipid bilayer is found. That is, in between two protein layers, a lipid bilayer is present.
স্যার সাইডেরটা দেখা যায় না।
In between two protein layers, a lipid bilayer is present. This is another characteristic. Next one. It's a living membrane. That means, so you have to, so it's found in all cellular organisms, thin and selectively permeable, made of protein, lipid, and protein. That is lies between two protein layers, and lipid bilayer is found. And lastly, it is a living membrane. So, sir, bolo. First one is sandwich model. By and Davison. First one is, what is called the sandwich model proposed by Danieli and Davison. And according to this model, according to this sandwich model, you know what it appears. I am showing you. Look. So this is actually one lipid layer. Now I am doing the other lipid layer. So it's a lipid bilayer. It's a lipid bilayer and just outer to which so this is the lipid bilayer. Is it clear now? This is the sandwich model proposed by Danieli and Dawson. This end is called polar end and these are called non-polar end. Actually, in one fatty acid molecule, suppose so this is actually a lipid is of phospholipid type. So phospholipid, do you know what is phospholipid? I am just making you understand what is this actually. You need to understand now. Do you know what is this? এটা কি বলতে পারবে?
স্যার এটা সোপ না?
It is a glycerol. You can call it trihydric alcohol. Then to which এগুলো কি fatty acid. There is to one glycerol molecule, 3 fatty acids are found to be attached. Now, যারা একটু মুছে দিই। Now it is a triglyceride. With the release of this OH and OH from here, eventually a triglyceride is formed. Now listen carefully. But it is a phospholipid. What is special about phospholipid I am showing you? For this one phosphate group, one fatty acid is required to be replaced by a phosphate group. That means, look at this diagram. Look here, a phosphate group is added. Now you can call it phospholipid. Now you can call it phospholipid. And this is what like this. Phosphate group is added to replacing one fatty acid to bind to glycerol to form. So now, what I say, look, this is the glycerol head and this is the fatty acid tail. One is glycerol head, the other is fatty acid tail. So glycerol head is here and it is hydrophilic. This polar end is hydrophilic, but non-polar end is hydrophobic. It is water loving, it is water hating. So polar end hydrophilic, non-polar end hydrophobic. Clear? On to the lipid. And another most important thing here, non-polar ends of both lipid layers are found to be directed towards the center. Found to be directed towards the center. This is the center. So center is actually hydrophobic. In both ends it is hydrophilic. So this is what the Danieli-Dassos model, but it has some limitations. What is the limitation of this model? The basic limitation of this model is that this model can't explain how water-soluble molecules can pass through it. As you know, the center is hydrophobic. Look one thing. Suppose if it is the two ends, here you will get the protein, the center is actually hydrophobic. As the center is hydrophobic, then how this membrane can allow water-soluble molecules to pass through it? This could not be explained by this model. The water-soluble molecules, how they can move through this membrane, is not explained by this model. Clear? Because here it is hydrophobic. How water-soluble molecules can move through? Is it clear to you?
Yes, sir.
Second one.
Yes, sir.
It can't explain the active transport of ions. Active transport, you know. For active transport, do you know what is active transport?
স্যার যখন একটা জিনিস নিজের এটিপি ইউজ করে নিজের এনার্জি ইউজ করে নিজের লোয়ার কনসেন্ট্রেশন থেকে হায়ার কনসেন্ট্রেশনে যায় তখন এটাকে বলে অ্যাক্টিভ ট্রান্সপোর্ট।
The movement of ions or molecules takes place against the concentration gradient with the expenditure of energy. The first two. Ions or molecules Look here. Movement of ions or molecules against the concentration gradient. It should be against the concentration gradient with the help of carrier. Obviously, carrier is required. Without carrier, there is no active transport. And with the expenditure of energy. But here you see, proteins are not even attached to the lipid. How these proteins can act as carrier to carry the ions? That's why this membrane can't explain what? The active transport. It can't explain how water or water soluble molecules can pass through it. It also can't explain how active transport occurs through it. So this is the basic limitation of this model. Robertson property. Jara to muche di. Eta muche di ra. Unit membrane model proposed by Robertson. And according to this model,
ইউনিট স্যার এটা কি ইউনিট মেমব্রেন?
unit membrane model.
আচ্ছা
What about this? So here again it is the lipid bilayer extended protein and again they are the polar end. Here you will get the non-polar end. Again it is the polar end. Here also proteins are not found to be attached to the lipid bilayer. You see, proteins are not found to be attached to the lipid bilayer. And again, so that the drawbacks or limitations found in the Danieli-Dobson model, same is also found here in the unit membrane model. So what is actually Robertson said, like cell membrane, all membranous structures present in cell are made of protein, lipid, protein. Robertson says that all, like cell membrane, all membranous structures present in the cell are made of protein, lipid, protein. And he described this protein, lipid, protein membrane as unit of all biological membrane or unit membrane. Are you getting me? That like cell membrane, all membranous structures found in a cell are made of protein, lipid, protein. Hence he described this protein, lipid, protein membrane as a unit of all membranes of the cell or all biological membranes or unit membrane. Is it clear to you? If someone has problem, then you can write it. According to Robertson, like cell membrane, all membranous structures found in a cell all cytoplasmic membrane लिखे दो. All cytoplasmic membranes found in a cell, all cytoplasmic membranes found in a cell are made of protein, lipid, protein.
Sir, repeat.
Like cell membrane, all cytoplasmic membranes present in a cell are made of protein, lipid, protein. Are made of protein, lipid, protein. Hence he described this protein, lipid, protein membrane. Hence he described this protein, lipid, protein membrane as a unit of all membranous structures, or as a unit of all membranous structures or unique membrane. All
কি বলেছে এখন? Say, hence he described?
Hence he described these protein, lipid, protein membrane as a unit of all biological membranes or all biomembranes, all biomembranes. Or unit membrane. Or unit membrane. Clear? Is it clear to you? The limitations that the Danieli-Davidson model or sandwich model is there, the same is found in case of unit membrane models. So then that's why the other model comes. এবার একটু মুছে দেবো এরকম, মুছতে হবে না হলে হবে না। আচ্ছা এদিকে পাওয়া যাবে কিনা দেখতে পারি, তিনটে একসঙ্গে থাকবো। Then he proposed the third one. Fluid mosaic model proposed by Singer and Nicholson. Singer and Nicholson proposed the fluid mosaic model. What about it? Look here. Again, it is the lipid bilayer. But the proteins are either found attached to the lipid membrane, lipid head or glycerol hull. It is found to be partially or fully embedded in the lipid bilayer. So lipid molecules, protein molecules are either attached to the, just slightly attached to the lipid membrane, or it is found to be embedded at different depth in the lipid bilayer. So these proteins called extrinsic protein and now you see this protein is called so you know there is a pore in the center so that this membrane can allow the movement of water and water soluble substances through it. Water and water soluble substances through it. Now here you will get something more. So you get here cholesterol in between the lipid molecules. You will get cholesterol. The role of cholesterol is to provide rigidity to the structure of lipids. Provide rigidity to the structure of lipids. Now you see here, similar polar and nonpolar ends are here. Now the extrinsic protein, which are just attached to the lipid bilayer, not entering into the lipid bilayer, they act as receptor. So you see, in this case, the extrinsic protein acts as receptor. They have to act as receptor, they have to receive the stimulus. And intrinsic proteins here act as carrier. That means it can now explain the active transport through this membrane. Here you see, as in between the pore is developed in the transmembrane protein, so that water and water soluble substances can easily through these pores. That means through these water and water soluble substances can easily pass through. So this is the most important thing, and this problem is resolved. Regarding the, you see here, one of the extrinsic protein you know is called spectrin. The example of extrinsic protein. Spectrin. Spectrin protein is actually extrinsic protein found in the cell membrane of RBC. So is it clear to you? Look at this clearly. DT to the cell membrane. Suppose this is a target, if I am going to draw it. এই হচ্ছে ট্রান্সমেমব্রেন প্রোটিন, দেখতে পেয়েছো?
ফাংশন কি?
এই যে actually no, there is a pore inside. So this pore, water soluble substances can pass through it. Because so the hydrophobic effect is not there. বুঝতে পেরেছো? Hydrophobic effect is not there. Clear? বুঝতে পেরেছিস? These are proteins which are fully embedded, and both ends are found outside the lipid bilayer. Both ends of this protein found outside the lipid bilayer. Clear? And there is a pore in the center of this transmembrane protein, or a passage found in the center of the transmembrane protein through which water and water soluble substances can pass through, because the hydrophobic effect is not there. বুঝতে পেরেছো তো? এটা বলা একটা মিনিট আসে। Most important thing here in this case or in fluid mosaic model, which may be compared with the protein iceberg in the sea of lipids. Now the question comes. Protein iceberg in the sea of lipid, and in this way it is actually explained. Protein, you know, the icebergs found to be floating in the sea, there some part is found to be embedded in the sea, under the sea, and some parts are exposed. That's why it is compared with the protein iceberg in the sea of lipid like this. And here the sea water is compared with the lipid, and the icebergs are compared with that of the proteins. Now, so actually it means lipid is a quasi-fluid layer. So lipid is a lipid is quasi-fluid in as lipid is quasi-fluid in nature in the cell membrane. So lateral movement of protein molecules takes place through it. As it is quasi-fluid nature, so lateral movements of proteins possible within the lipid bilayer. So in this way fluidity is maintained. I am repeating it again. According to this theory, as lipid is quasi-fluid in nature, or semi-fluid in nature, so protein molecules show lateral movements within the layer. The protein molecules show lateral movements within the overall bilayer. Are you getting me? Proteins can move laterally. How is it possible? As lipid is quasi-fluid, it is not a static hard membrane. As it is a quasi-fluid membrane, so protein molecules can move laterally. Protein molecules can move laterally within this layer, and hence actually in this way the fluidity of membrane is maintained. Are you getting me? Due to the quasi-fluid, semi-fluid. It is floating, not it is static. It is not a hard one. Semi-fluid or quasi-fluid in nature. Which one? The lipid bilayer. As lipid is quasi-fluid, so the protein molecules can show lateral movements. That means protein molecules can move laterally. protein molecules embedded within this lipid bilayer can move laterally. Are you getting me? How these proteins can move laterally? As this lipid is a quasi-fluid layer. As this lipid is a quasi-fluid layer, that's why the proteins can move laterally within this lipid bilayer. So, intrinsic. Are you getting me now?
Yes, sir.
And in this
Yes,
way, it's called, this ability of movement is considered as a measure of fluidity. Lipid molecules are also shown to move also, obviously. Lipid molecules can move laterally also. But proteins can also move laterally within this lipid bilayer. So, is it clear now? As lipid is a quasi-fluid in nature, protein molecules move laterally within this lipid bilayer. Clear? And these abilities measured as a measure of fluidity. Is it clear up to this? So you see, is it clear the differences between these three layers? এই তিনটা মধ্যে ডিফারেন্সটা ক্লিয়ার হয়েছে?
Yes, sir.
Okay, that's fine. Now you know, as I said earlier, the example of extensive protein I gave you is the permease, is pectin in the RBC. And lot of intrinsic proteins are found. Some of these are actually act as, you know, the enzymes. One intrinsic protein acts as enzyme called permease. পরীক্ষা আসে Permease is a protein present in the cell membrane, acts as enzyme. Can you tell me, how is flexibility? Write down, please. How is flexibility? How is flexibility and dynamism of cell membrane maintained? কি হবে তাহলে? What I said earlier. As proteins can, the movement of lateral movement of the proteins through the quasi-fluid lipid bilayer, movement of the proteins present in the cell membrane, lateral movement of the proteins within the quasi-fluid lipid bilayer maintain flexibility and dynamism of the cell membrane. Are you getting me? Lateral movement of the proteins within the quasi-fluid lipid bilayer maintains the flexibility and dynamism of the cell membrane. So, remember this thing, the flexibility of dynamism is dependent on the, basically based on the, what is called lateral movement of protein molecules within the quasi-fluid lipid bilayer. Remember this thing. Likhe niyecho? Dynamism, it can change its shape.
Sir, তাহলে তো flexibility হয়ে গেল না?
Flexibility means actually it is flexible. It is not a static one. Suppose these are static, they are not changing their position. বুঝেছো?
Yes, sir.
হ্যাঁ, স্যার।
Now, next comes to that. এই জায়গাটা একটু মুছে দিই, একটু মুছতে হবে আসলে উপর জায়গাটা। একটা জিনিস দেখাতে হবে এখানে। একটু এটা। Now here you see some glycolipids or glycoproteins.
স্যার পারমিয়েসটা কি বলেছিলে?
একটু দাঁড়াও বিশ্বাস করে নিয়ে আসি। Look here. To these protein and lipid molecules in the outer side, some oligosaccharide chains are found to be attached. Some oligosaccharide chains are found to be attached to this lipid and protein layers. So, if you can call it glycolipid. So in this way, actually it is an oligosaccharide chain attached to, you can write here, this is the oligosaccharide chain attached to protein, hence it is called glycoprotein. And here also you can write this is the oligosaccharide chain attached to lipid and hence it is called glycolipid. So you will get glycoprotein and glycolipid structures which constitute the glycocalyx or cell coat. So what does it form actually then? They form the
Sir,
wait.
Sir.
Wait. Glycocalyx or cell coat. This glycoprotein and glycolipids forming the glycocalyx or cell coat outer to the cell membrane. হ্যাঁ বলো।
হ্যাঁ না এটাই জিজ্ঞেস করা ছিল, গ্লাইকোলিপিড আর গ্লাইকোপ্রোটিন কি গ্লাইকোক্যালিক্স হয় নাকি?
বুঝতে পেরেছো তো? So in this way you see a protective layer is also formed around it. Basically this function is the recognition center, site of attachment. His role is to act as most important recognition center. That means it's to recognize which are to enter the cell and which are not. Then comes to the site of attachment. And the site of attachment, where the molecules before entering into the cell they have to be attached here. So this is what about the fluid mosaic model as proposed by Singer and Nicholson. Is it clear? So whenever the extrinsic protein attached to the phospholipid head by a covalent linkage, that means I am showing you So here you will get a covalent linkage with the phospholipid head. Remember this thing. Sometimes some extrinsic proteins are found to be attached with the transmembrane protein. So sometimes extrinsic proteins are found to be attached to transmembrane proteins. So here extrinsic protein attached to transmembrane protein by non-covalent linkage. So two types of linkage found here. One is covalent linkage in between the extrinsic protein and the phospholipid head. Other is the non-covalent linkage between the extrinsic protein and transmembrane protein. So this is what about the fluid mosaic model as discussed by Singer and Nicholson. Clear? After all, the questions come, what should be the function of cell membrane? Now the most important question comes now. Most important, after studying cell membrane in different aspects, different models are discussed, we know its location, then I would like to know what is the basic function of cell membrane. First, most important I am telling you, its main role is to separate the cell from the external environment. Secondly, can you tell me? It provides a particular shape to an animal cell. Thirdly, with the help of cell membrane, a cell can engulf solid particles, or a cell can drink liquid. That is, pinocytosis and phagocytosis is possible. Cell membrane can give rise to some organelles like endoplasmic reticulum.
Sir?
বলো
স্যার একটু লিখে দাও না, কথাগুলো আসছে না।
আসছে না? আচ্ছা ঠিক আছে
স্যার বুঝতে পারছি না।
ঠিক আছে। No, a lot of functions are there. It can give rise to some organelles also. So this is all about the cell membrane. Now we have to discuss some basic points here. You need to understand now. Passive transport. Look at these three. Active transport, facilitated diffusion, and passive transport. Look one thing. In passive transport, movement of ions or molecules takes place along the concentration gradient without any carrier or without expenditure of energy. So here, so whenever movement of ions or molecules takes place without the expenditure of energy and without any carrier and along the gradient, that means from higher concentration to lower concentration, then it is called passive transport. So in passive transport, the ions molecules move from higher concentration to lower concentration, no carrier is required and energy is also not required. Then you can call it passive transport. In case of active transport, in case of facilitated diffusion, what happens here? Movement of ions or molecules movement of ions or molecules takes place along the concentration gradient, similar to the passive transport, but with the help of a carrier and without expenditure of energy. So in two parameters, it is similar to the passive one, that movement occurs along the gradient, from higher concentration to lower concentration, and here no energy is required. So in these 2 points, they are similar, but they are different in the point that the carrier is required in facilitated diffusion. Carrier is required in facilitated diffusion. This is the point of difference. In case of active transport, what happens? Movement with the help of a carrier and look, this is the active. So active transport is found similar with the passive diffusion in the point that in both carriers are required, but different from that in 2 points. One, energy is not required in facilitated diffusion, but energy is required in active transport. movement occurs along the gradient in facilitated diffusion, but movement occurs against the gradient in the active transport. Now you see that whether clear to you that what are the actually the basic difference between active transport, facilitated diffusion and the passive transport. Dekha now it too. Clear kina? Actually it is also in different chapters are here, but as it is here, I am to say about it. Osmosis and diffusion. Do you have any idea about osmosis? Anyone can say, please? The semi-permeable membrane. Now you see here two solutions are there separated by a 0.2 solution of different concentration. Look, 0.2 solution of different concentration are separated by a semi-permeable membrane, and movement of solvent takes place from its higher diffusion pressure towards lower diffusion pressure region. Then it is called osmosis. Remember this thing, movement of solvent takes place where there is higher diffusion pressure of solvent is there towards the region where the diffusion pressure of solvent is less. Diffusion pressure is maximum in case of distilled water. So with the addition of solute, gradually diffusion pressure decreases. So you will get higher diffusion pressure of solvent where amount of solute is less. That means in B, diffusion pressure of solvent is more in comparison to A. Clear? So, movement of water solvent takes place from B to A. This is osmosis. When two solutions of different concentration are separated by a semi-permeable membrane, movement of solvent molecules takes place from the higher concentration of solvent or higher diffusion pressure of solvent towards the region of lower concentration of solvent or lower diffusion pressure of solvent till equilibrium is maintained, then it is called osmosis. দেখো বুঝতে পেরেছি কিনা? In osmosis, movement of solvent takes place from the higher concentration of solvent, not solution. Higher concentration of solvent means where the amount of solute is less. Maximum concentration of solvent is found in distilled water. So higher concentration of solvent means where amount of solute is less, and lower concentration of solvent means where amount of solute is more. That means concentration depends on diffusion pressure. Diffusion pressure means ability of the solvent to move. So diffusion pressure decreases with the addition of solute. The more you are going to add solutes, the diffusion pressure of solvent decreases. So you can call it low concentration of solvent. When the amount of solute is less, obviously diffusion pressure is in that case more. The maximum diffusion pressure found in distilled water. So in this we can say when two solutions of different concentration are separated by a semi-permeable membrane, movement of solvent takes place from a region of higher diffusion pressure of solvent towards the region of lower diffusion pressure of solvent, or from a region of higher concentration of solvent towards the region of lower concentration of solvent. But don't forget it's not the solution. Clear? Osmosis? এই মুহূর্তে তুমি সলিউট অ্যাড করবে, পজিটিভ ডিফিউশন প্রেসারটা কি কমবে না বাড়বে? কমবে, তাই তো?
Yes, sir.
হ্যাঁ, so where there is more solute means less diffusion pressure.
Yes, sir.
That's why the movement of solvent takes place from a region where there is less solute towards the region where there is more solute. ঠিক আছে?
Yes, sir.
And then this is called osmosis. Clear? And diffusion actually is also osmosis may be termed as diffusion of solvent. It is called diffusion of solvent. বুঝতে পেরেছো কি বলেছি? It is called diffusion of solvent. Why? Because in diffusion, movement of molecules takes place from a region is higher concentration towards the region of its lower concentration. Here also, water molecules move from the region of the higher concentration of water towards the region of lower concentration of water. Clear? That's why osmosis is also a type of diffusion, but it is only the diffusion of solvent. But you know, diffusion takes place from actually solid to gases, liquid to liquid, solid to liquid. A lot of diffusion is there. Whenever it is liquid to liquid, then it is called osmosis. বুঝতে পেরেছিস?
Yes, sir.
Okay. So that's all for today. আজকে আমরা এখানে শেষ করছি। Next আমরা